Good afternoon and welcome back to our second live Q&A session of the day. During this session, we're going to have an opportunity to hear from some of the researchers behind the AMMF-funded research. We're joined in this session by Molly Wilson, Dr. Stephen Griffin, and Professor Matthias Avila. And this session will be chaired by Dr. Christopher Wadsworth, who I'd like to hand over to.
Thank you very much, Gary. Thank you to all three of the colleagues joining me now to discuss the really impressive bodies of work that have been presented to us just now. I think it reflects obviously very well on them and also on AMMF for supporting this sort of work and the commonalities of this being translational work where there's a clear focus on trying to work out the pathology of this disease, but also develop targets for new treatments. Is obviously very, very important. So without any further ado, we will move over to some questions, and I'll start by putting a question to Molly, and maybe I'll try and keep it roughly in that order. Please do keep submitting your questions in the messaging system so I can put it to our speakers. So Molly, the first question I've had submitted for your attention is, thank you very much for your talk. Have alterations in NF2 been found in CCA tumours before? So I presume human excision tissue. And I'd supplement with that do we see increased rates of cholangiocarcinoma in patients with neurofibromatosis?
So I can answer the first part of that question. So in our computational screen, where we looked at the genetic data of 200 cholangio patients, so 2 of those patients had loss of function mutations in NF2. And then in terms of neurofibromatosis patients developing cholangio, I haven't actually looked at that specific link, so that would be something I need to examine closer.
Yeah, I think they've got higher rates of GI cancer, but I don't know whether.
Yeah, I'm not sure about specifically cholangio.
Thank you. So a question now from Steve, and it feels a bit weird just reading Steve's question out, but very nice talk, Molly. Merlin NF2 regulate hippo signaling, as you mentioned. Did you look at YAP-TAZ activation?
Yeah, so this was actually very interesting because NF2 is such a prolific activator of the hippo pathway, right? So I did some staining for YAP-TAZ in some of our tissue and found that it was all nuclear, so that would be indicative that the hippo signaling is off. And also then I looked at a lot of the hippo signaling pathway components in the RPPA, like the proteomic analysis, and we didn't get any change in those either. And I mean, NF2 is at the nexus of all of these different signaling pathways. So we also had greatly increased EGFR signaling, but again, when we looked into that in greater detail, that was it was greatly increased in the hepatocytes, but not in the cholangiocytes. So that's how we kind of came to settle on the Wnt beta-catenin and AKT signaling.
So sorry, Molly, you're saying that the YAP and the TAZ were in the nucleus?
Mm-hmm.
So that would mean that they were active, and so when hippo is inactive, then the YAP and TAZ goes to the nucleus, and so you will be getting transcription driven by YAP and TAZ in that scenario.
Mm.
Yeah.
Food for thought there. Thank you, Steve. Question now for you, Stephen. This is a question from Scott Waddle. Interesting talk. From what I understand, you spoke of some in vivo evidence to show that LPCs are infected with hepatitis C, become mature cholangiocytes. Are there lineage tracing experiments in vivo to confirm viral infection of LPCs, non-cholangiocytes, causing differentiation to mature cholangiocytes?
No, there isn't. So it's an indirect inference, really. We can't do those experiments in mice because adapting hepatitis C to mouse models isn't really viable. It requires lots of immune suppression and modification of the mice, and those experiments just haven't been done, unfortunately. The data I showed was from an old paper where we could detect hep C antigen both in the bile duct and in fact secreted in bile. And because we know that mature cholangiocytes lack the surface receptors that hep C requires to start an infection, you cannot infect mature cholangiocytes with hepatitis C. We infer, therefore, that they must have either transdifferentiated from a hepatocyte that was infected or indeed been generated by a progenitor cell. And because of the extent of the antigen that was found there, we believe that that's the case. And of course, our ex vivo model supports that.
And Steve, I'm sorry if this was fully covered in your talk. I guess I didn't grasp if it was. In terms of the corollary with HCC, do you see that this Hippo pathway, is that important there? Have you demonstrated the same in HCC, and has that been looked at as a therapeutic target there?
Well, people are developing Hippo pathway. Well, it's not right to say Hippo pathway inhibitors because you don't want to inhibit the Hippo pathway as such. You'd want to activate the Hippo pathway in order to inactivate YAP and TAZ. So it's a bit of a strange signaling period. And yes, drug companies, pharma, are definitely targeting that. It's very difficult to get an assay to measure that effect on gene expression. And actually, we're talking with people about our system to do that. But yes, it's involved in the route towards an HCC as well as the route towards a CCA, because starting from that same fundamental start point that we're modeling, the Hippo pathway is involved with, preventing that terminal differentiation coming out of cycle that you would expect to be involved in both. And it's probably the external mediators of either hepatic differentiation towards hepatocytes or cholangiocyte differentiation that modulate where that may or may not end up.
Okay, thank you. So I'm just going back to the chat here, and okay, so there's not many chat questions coming here, but I've got some previously submitted questions here. So Matthias, if we could move to you in the sort of the experimental work you've done on this novel agent CM272, you showed some very nice pictures showing the reduction in the volume of the tumor where you introduce in the mice where you introduced this agent. Did you actually see regression of the tumor, or was it that you just saw that they failed to develop the tumors at the same rate as or instance as the untreated group?
Well, we tested, as you may have seen, in different in vivo models. In the PDX model, we did see a very strong effect, and there was a regression. The tumors did not grow at all. They even shrink. In the xenograft using human cell lines, we saw an inhibition of the growth. Okay. In the in vivo model of endogenous tumorigenesis on a background of inflammation and fibrosis, and there we saw quite a remarkable effect of preventing the progression of the tumor, and the lesions were quite diminished. Mm-hmm. So every model gives you some information that is complementary.
Yeah. Okay, thank you. And has this agent been used in any other conditions or experiments on any other tumors, either in humans or in animal models?
Yeah, we've done some work in hepatocellular carcinoma, and it also works very, very nicely. And in some other collaborations, we have tested in hematological malignancies, and as well, the effects were quite visible. And there is another work already published on bladder carcinoma. So I think there are several tumors that are maybe sensitive to this agent, and so it's and the toxicity we've observed. The lack of toxicity is quite interesting, because you think that you are using not so specific, targeting such a pathway that is controlling so many genes. You should see some toxicity, but we haven't. We've administered it to mice for over a month. We've done some experiments on human precision cut liver slices. You can keep these liver slices for almost a week, and we treated with the compound, and we did not see any toxicity. On the contrary, we saw inhibition of fibrosis. So I think so far, we are trying to move the molecule forward to clinical trials. Yeah.
Sounds incredibly promising with that sort of that signal from multiple different studies on different cancers that it's proving potentially effective. Great. So another question for you, Matthias, from Professor Diego Calvisi. Dear Steve, an excellent presentation. Have you looked at the notch pathway downstream of YAP-TAZ in your model?
Not specifically. We've done some microarray studies in cholangiocarcinoma cell lines, and indeed we saw some effects, target genes, and some indications that this pathway could also be somehow modified or inhibited by the compound, but we did not pursue it further so far.
I'm sorry, and I've really confused the situation by asking you a question that was directed at Steve, Matthias, so you did incredibly well answering it. Steve, would you like to respond to the same question? That was entirely my fault. So, Steve, an excellent presentation. Have you looked at the Notch pathway downstream of YAP-TAZ in your model?
We certainly are going to, because Notch itself is dysregulated by the Hep C infection, as are many downstream effectors there. And it's quite interesting, following on from Matthias's talk, which unfortunately I didn't get to see half of because we were here. The way that the virus seems to have this hit and run effect on the cells, and why we think it's quite useful, is two ways. One, the viral proteins are kind of a Swiss Army knife and dysregulate multiple signaling pathways in the sort of cytoplasm. But there's also this epigenetic remodeling in the cell, which is causing dysregulated gene expression. And that's probably where this imprinting idea comes from. And actually, people are looking at epigenetic modulators, such as Matthias is talking about, to try and restore those adverse phenotypes in the circumstance of the virus infection. But yeah, certainly Notch and TP63 pathways are clearly involved in these procedures as well.
Thank you, Steve. So, Matthias, another question for you. Your in vitro and in vivo data for your CM272 compound look really promising. Are you surprised that dual inhibition of DMMT and G9A wasn't more toxic in the healthy control or the mice treated?
In my previous answer, I alluded to this point. I think, yeah, I mean, we started working with this molecule about 4 years ago, and in initial studies, we were afraid that it could, induce some significant toxicity, because it's it's targeted, very specific molecules, but these enzymes are, part of, the regular work of the cells. But as I said, I mean, we did not see, we measure, liver toxicity, kidney toxicity, hematological toxicity. The mice did not lose weight. And as I said before, we had access to these precision cut liver slices from, patients and transplant liver. And on the contrary, I mean, we even in models in which you have already ongoing liver injury, you could expect to be, more sensitive to damage. We had the opposite effect. I mean, we also studied the effects of this molecule in several models of liver fibrosis in mice, and again, I mean, we saw anti-inflammatory responses and anti-fibrotic responses. So, I mean, so far, the regulatory toxicity has not been done yet, in order to get registry as a new drug, but from the models and experiments we've done in our lab and others, we have not seen that.
Yeah, and I guess we have to be cautious because these are mouse models, but it's promising, yeah. Very exciting and promising today. Matthias, another question for you from Diego Calvisi. Sorry, the questions keep moving. Have you seen signs of redifferentiation in the remaining cholangiocarcinoma lesions?
We have not measured that specifically. We've done this analysis in vitro cell lines where, we could restore the expression of genes associated with the mature and differentiated cholangiocyte like SOX17. And also we saw the induction of genes that are also expressed in normal cholangiocytes, metabolic genes, and in a way that are shared also with hepatocytes, because there is also this idea that if you can differentiate these cholangiocarcinoma cells towards a more hepatocytic, profile, you also reduce their malignant behavior. But we have not measured that specifically in the tissues.
Okay, thank you. And Stephen, a question from you from Dr. Kai. Wonderful presentation. Does HCV infection induce cholangiocyte differentiation to stem-like cells?
So reversing terminally differentiated cells. We've not done those experiments, I'm afraid, Dr. Kai, in cholangiocytes. Certainly, Hep C prefers to be in a kind of mid-stage of differentiation in our hepatocyte models, and people have shown that infecting mature hepatocytes ex vivo with hepatitis C does induce an EMT type transition and dedifferentiation. So we would predict Again, as I mentioned before, we can't infect mature hepatocytes, cholangiocytes with hepatitis C because they lack the surface receptors. However, what we could probably do, which we might be able to do with Alex's stem cell system, is to reverse differentiation in those once we've got the virus in at the hepatoblast stage. So kind of go that way and come back maybe. But there are other tricks we can play, but we would predict so, yes.
Lovely. Thank you very much. Well, thank you very much to the panel. We've reached the end of our allotted time now, and I believe it's time for the lunch break. I hope I haven't got that wrong, and send everyone off on their lunch. Thank you very much indeed to Stephen, to Matthias, and to Molly for their superb presentations and the tremendous work they're doing in this field. And thank you very much to AMMF and their supporters for all the work they've done to support this. Over to you, Gary.
Second what you say. Thank you, Chris, for chairing this session, and thank you to the AMF researchers for your time and commitment. Speak to you soon, and thank you for joining.
Thanks, everyone.
Thank you.
Thank you. Thank you, everybody.
Bye.